for agreeing to come and give a presentation, which is very relevant to our interests. She's the CEO and the founding member of PharmaPRO Consulting. Her company specializes in clinical pharmacology and regulatory affairs and PKPD data analysis. She got a PhD from LIU, Long Island University, where I graduated from. She's the chair of the Society of Pharmacometrics, a local chapter. She is a chair for the ASCPT, American Society for Clinical Pharmacology and Therapeutics. She has received many awards from Pierce, Eli Lilly, AstraZeneca, and Inclone, and other places for her contributions. She conducts low-cost workshops all over the world on clinical pharmacology and PKPD modeling. She particularly supports companies for expedited FDA approvals, pediatric clinical pharmacology, and mathematical modeling for drug development. I think I'll take up more time if I start going through all her expertise and experience. I'll let Aruna take it from here.
Thank you, Nat. Thank you for the kind introduction, and thank you for the opportunity to come and present and share some of my experiences with your students as well as scientists in the organization. It's a pleasure to be here. I have tried to keep my presentation more practical, and I will be covering some pieces of data and which also covers some of my personal experiences when it comes to drug development and overall regulatory approval. So let me share my screen here. So the topic that I'm going to be talking about is what is model-informed drug development and how can it be used effectively to save time and money in drug development. As Nat mentioned, that I have worked in the industry for over 27 years, and I specialize in clinical pharmacology and mathematical modeling. So today I'm going to be covering some of the basics of model-informed drug development, also known as pharmacometrics, also known as MIDD. The outline of my talk today is I'm actually going to zoom out. This is something that you might already know, the various phases of drug development, just to get us up to speed. And I will also cover in terms of how much time does each phase of drug development take. Then I'm going to talk about the FDA drug approval process. I mean, this could be applied to any other regulatory agency around the world as well. Because we are in the United States, I will focus primarily on the FDA. And then we will talk about what does it take or how much does it cost in drug development to bring a new medication into the market. And this is data that comes from an analysis that was done by a team at MIT. And then I'm going to take a deeper dive into what is model-informed drug development, or MIDD, the applications of model-informed drug development. What is the regulatory perspective when it comes to decision-making in support of model-informed drug development? Then I will give a high-level overview of the current decision-making process or how the clinical drug development has expanded to include model-informed drug development, and we will actually look at some current data. Then I'm going to cover a few case studies, that is real-world evidence based on my own personal experience on how model-informed drug development application actually led to saving huge amount of time and also in terms of revenue when it comes to the drug development. Let us take a broader overview of the drug development process. Now, many of you may have seen this slide earlier. So this slide is basically presenting the broader view of drug development process. And it also highlights few important key pieces. One is you're looking at what does it take, from going from pre-discovery to approval process. And here we are also looking at the time scale in terms of the drug approval process. So we move from discovery to preclinical to clinical. And again, the clinical is divided into phase 1, phase 2, and phase 3. Then we look at NDA or a new drug application submission to the regulatory agency, post which The FDA would take a certain amount of time to review the application that generally ranges from half a year to a year, post which the FDA chooses or decides based on whether FDA wants to give an approval or not. Then we move into the post-marketing phase of the drug development. And I also want to bring your attention to what is the attrition rate when we are looking at pre-discovery. We generally move from something like 5,000 to 10,000 compounds that are explored. So by the time it gets to preclinical stage, maybe 250 of them go through the preclinical stage funnel, out of which maybe 5 make it to phase 1 clinical trial. And as you can see, out of the 5, maybe 1 gets to the NDA submission stage. And we will discuss this a little bit more in length in our future slides. Also looking at what is the timeline that the drug development process takes. So moving from pre-discovery to IND, it takes about 3 to 6 years. IND, also known as investigational new drug application, that is submitted to the regulatory agency or the FDA. Post IND, it takes about 6 to 7 years for a drug to go through various stages of clinical drug development. After which, if we do have acceptable data, then we go through a new drug application process or a submission to the regulatory agency. So let's move further on, and let me actually bring your attention to a few important key numbers. I think we talked about how do we save time and money using model-informed drug development. Before we understand that, let us look at what does it take. What is the current data showing? So the plot up top here represents the success rate or probability of success for a compound to move from phase 1 to phase 3 to approval. So the current data, and this, as I mentioned, was an analysis conducted by 3 different groups. And the first group here is talking about the success rate, right? So this is the percent. So there are about only 13%, or rather 13.8% of drugs that start in phase 1 actually get to phase 3 and approval. So as you can see, a very high attrition rate. Moving on to the next figure on this slide. This covers how much is the time that an average drug takes to go through various stages of drug development. So here, this plot actually represents 10-year data from 2010 to 2020. And as you can see, this is the number of years that a drug takes to go through the various stages of drug development. And on an average, this analysis suggests that it takes about 9 years or more for a drug to go through the drug development process. And as you may have already thought, what is this representing? So this, the third figure here on the slide, is actually representing the cost. How much does it cost to bring a new medication into the market? As you can see, that it takes about $985 million for an average compound to go through discovery through approval. So close to $1 billion is what is spent in R&D for the drug to go through the approval process. All these numbers are pretty staggering. So let's look at the same thing that we were looking at in terms of drug development. I did not put any definitions per se, because the definitions you can always get from the internet. And that is not our focus here, right? What is model-informed drug development? It is primarily what you can kind of understand, that there is a mathematical model that represents the pharmacokinetic, pharmacodynamic, physiologically based model. So basically a mathematical equation explaining to us how the drug is behaving. That is what is a model. And that model is developed based on real-time data, and sometimes based on data that was made available either in the preclinical setting or in the clinical setting. And that information is what is used to make certain projections. As we are going through the presentation, you will actually learn more about it. And these are the various regulatory decision support processes that happen through the drug development and approval process. So what are the various phases of what happens in a model-informed or MIDD process? So a model is something that is developed, or mathematical model is something that is developed way early on in the drug development phase. And this could start as early as in preclinical setting. And as the drug is actually moving through the various stages of drug development, we actually utilize this cycle or the circle that talks about as you have some knowledge, you capture it to create a best model, and that is used to make certain predictions. And based off of those predictions, when you get real-time data, you go back and re-optimize this model. And it's a cyclical process, right? More data, more information, more optimization of that model. When can one start using model-informed or developing a model? One can start as early as in pre-discovery stage. And as you're moving from test to a preclinical or an animal setting, and as you're moving from the animal setting to a clinical setting or in humans, all of this data is going to be used to keep optimizing that model. So the application of model-informed drug development. Here, this slide actually represents the various phases that this can be used for. I think we briefly touched upon the PK, or that is pharmacokinetic analysis, the population PK analysis, PBPK is another setting, exposure response analysis, disease models in clinical trials, QSP modeling, in silico clinical trial simulations, and many others. And what stage of decision-making does a model-informed drug development process help? It can start as early as preclinical. It can be used for first-in-human dose predictions. It can be used to design better clinical trials. It can be used for pediatric drug development. It is also used to understand the drug's behavior in different settings. You can actually tease out if there are differences between male population versus female population. Does age have an impact on the dose at which the drug should be administered? It can also be used to design clinical trials for pediatric subjects. It is like we have data in the clinical setting wherein the drug was used in adult population. Based off of that, one can make predictions on how it would behave in pediatric subjects. And sometimes it can even be used for geriatric subjects to see if we need to conduct anything when it comes to dose optimizing, when we are looking at special disease condition, different metabolism profiles of the individuals, do they play a role, etc. So what is the regulatory stand when it comes to model-informed drug development or the utilization of these state-of-the-art technology? Some of you might know this. So there is a Prescription Drug Act that the FDA has put forth. And within that, the FDA has clearly given a strong position to model-informed drug development practices to be used in all drug development practices. In fact, there are guidance documents which talk about the use of model-informed drug development practices. It also covers how and when to use it, what are the best case scenarios. And often FDA actually expects that a pharmacometric analysis has been conducted in various stages of drug development. Sometimes when the sponsor or the pharmaceutical company interacts with the FDA would request for this data. And if they don't have that data, they might guide them on what needs to be done and how they can utilize the state-of-the-art technology to help them in dose and regimen selections, etc. So taking a broader view of how is the regulatory decision-making changed in the past few years. So here is, the information in terms of PBPK modeling or drug-drug interaction trials, and how you can see that these are the different therapeutic or different populations in which it has been used. And over the years, you can see the number of applications in which the PBPK modeling is being used has increased exponentially, which is definitely a great sign, which also means that this is a growing field and it will continue to grow over the years to come. So if any of you are considering taking on expanding your knowledge in model-informed drug development or pharmacometrics practices, or apply these understandings in your master's or PhD thesis, it's a great time to do so. So, taking a small pause here, just wanted to see if there are any questions. So moving on. So let me actually cover a few case studies. And these are real-time case studies, and I'm actually going to be covering some of the experiences that come from my direct involvement in drug development and how pharmacometrics or MIDD or model-informed drug development played a key role. So the case study one that I'm going to be covering is for one of the questions that we are trying to address. And the question was, what would be the dose or dosing regimen that we should be recommending for an ongoing phase 1 clinical trial? So what we wanted to look for is how can we make a well-informed decision in terms of what is the dose or dosing regimen. So this was a drug used for treatment of oncology. So the approach that we took was we combined all the data that we had, the clinical trial data as well as preclinical data for this particular compound. And then we looked at what would be the appropriate dose or dosing regimen to improve patients' compliance or improve what would be helpful to the patient is what we were looking at. And we've also wanted to address how can we design a better clinical trial. So the objective of that was obviously the appropriate dose. The most important piece was what are the appropriate sampling time points? Again, we don't want to, here, as I mentioned, this was an oncology drug. So in case of an oncology clinical trial, the clinical trials need to be conducted in patients, which also means that these are terminally ill patients. So we want to minimize the number of blood samples that we collect from these patients. And we also wanted to look at what would be the appropriate number of patients to be included such that we get enough evidence to support our next clinical trial. I think we kind of mentioned this, but I'm just going to say it one more time. So the overall objective was reduce the number of doses that we test in the clinic. Second, reduce the number of blood draws from these patients and minimize the number of patients, however, focusing on getting maximum output or maximum data from the minimum number of patients. So what was done was that we pooled all the data that we had and we created a model. And that model was used to run some simulations. And here we are, the green line here represents the simulated data and the dots represents the real-time value from a clinical trial. So here we were looking at, are we optimizing the model and is the model really predicting real data as it should be? So what we compared was if the drug was given at 10 milligram per kilogram every 2 weeks, how does the real-time data, compare when we are putting it against the model. As you can see, the model is predicting very well how the real-time data, what the real-time data was suggesting, which meant that we had good confidence in the model. And this model was used to run simulations at various doses. And before I move further on, maybe I'll bring you to the previous slide to talk a little bit more about what are these pink lines representing. In this particular case, we actually had data from a mouse xenograft study that was conducted, which suggested that these two would be the minimum efficacious concentrations that would be required for us to show the necessary response. Now, that being said, as you can see, that when 10 milligram per kilogram dose was given at every 2-week dosing regimen, we were actually not above these two thresholds that we wanted to be. Now, that could be one of the reasons why 10 milligram per kilogram did not show us the response that would be the appropriate response for the drug. So when we ran simulations, we were actually focusing on patient compliance. As I mentioned, we wanted to make this easier on the patients, and these were oncology patients. So this drug is actually administered in combination with a chemo agent, and that chemo agent is given at every 3-week dosing regimen. So to help the patients and to also help the efficacy of our drug, the best combination would be to give it at the same dosing regimen as the chemo agent is given. So we were looking at every 3-week dosing regimen to match the chemo. So we ran simulations at various doses at 15, 20, 25, and 30 milligram per kilogram every 3 weeks. This drug was a monoclonal antibody, so it was a protein. And based on the information that we had thus far for that compound, a dose of 25 milligram per kilogram is a pretty high amount of protein being administered to a patient, and that to an oncology patient. So a 25 milligram, as you can see, 25 was a dose that was kind of giving you a threshold minimum concentrations that were predicted to be efficacious. But that also meant that we are actually injecting a huge amount of protein to these oncology subjects. So based on this data, let me actually take you a few slides back, right? So what did we eventually come up with is the dose that would work, a dose that could be given at every 2 weeks or every 3-week dosing regimen. And we also wanted to look at how could we collect the PD or biomarker data, which would help us support in designing a right dose for the phase 2 study that would be the next study to come forward. Eventually, based off of the simulations, we actually determined the dosing regimen that would work best. And those doses and dosing regimens are the one that you would see in the case study 2 moving forward. So here is where I introduce the name of the compound. The name of the compound was Olaratumab, or 3G3, which is a fully human IgG1 monoclonal antibody. And this was directed platelet-derived growth factor receptor alpha. So that was the target that we were looking for. In order for us to determine the right dose and dosing at exposure response, that is a PKPD analysis to support the dose and dosing regimen for phase 2 clinical trial. So based off of those simulations that you saw earlier, and this is again, I'm just talking about, if you're interested to learn more about it, you can actually go to these two publications wherein we have covered the modeling project that was done and what was the dose that eventually was used in the clinical trial setting. So based off those simulations that you saw earlier, we recommended 3 doses to be moved forward into the clinical drug development. And the 3 doses one was 10 milligram per kilogram every 3 weeks. However, the drug, as you saw earlier, when given as every 3 weeks only, we were not reaching the concentration above which it would be efficacious. So we made a slight modifications or an innovative dosing approach, which was to give 10 milligrams on day 1 and day 8 of the 21-day dosing cycle. So the suggestions were to use 10 milligram and 15 milligrams in this dosing cycle, that was day 1 and day 8 in a 21-day dosing regimen, and then to use 20 milligram every 2 weeks, that was at a 14-day dosing cycle. And let me actually bring your attention to these slides again, right? So the blue line here actually represents the mean change from baseline for the PD biomarker, and the orange line here represents the PK concentrations over the period of time. And as you can see here, when you actually move from 10 milligrams to 15 milligrams, you would see that there is a 40% change from baseline. And when we are looking at 20 milligram every 2 weeks as well, you do see a 40% change from baseline. So based just on the PKPD comparison, what we see is maybe 15 milligram day 1 and day 8 and 20 milligram every 2 weeks may be appropriate dosing regimens to move forward. So how did this help in influencing the decision making? As you saw earlier, we were, again, because of proprietary reasons, I cannot show you the PKPD model that we developed. However, you're welcome to go to those manuscripts that I mentioned to learn more about that. So we did develop a PKPD biomarker analysis based on the simulations, and that was used to suggest the dose and dosing regimens to move forward for the phase 2 clinical trial setting. Based off of this, we actually conducted an exposure response analysis that was completed by the end of phase 2. And that data was eventually used to apply for a breakthrough therapy designation application. So let me actually take a step back and quickly share with you what is a breakthrough therapy designation application. A breakthrough therapy designation application is an application when the sponsor or a company goes back to the FDA at the end of phase 2 and provide enough evidence to support a special application approval. That is, in which case you actually can request that the drug gets approved under the special category, wherein only phase 1 and phase 2 data is used to support the overall approval of the drug. Doing so, one needs to meet certain criteria. One important criteria is that you do see that efficacy. You have done the work to support dose optimization, and you have also provided enough support to prove that the drug is safe and this is an unmet medical need. And in this case, the FDA has to give a response within 60 days, whether your request was approved or not, or whether the sponsor or the company needs to conduct more clinical trials. So because of all this analysis that we did, and we have robust data to support, this drug eventually was approved under the breakthrough therapy designation by both the EMEA as well as FDA. So saving about 5 years in Olaratumab drug development process, which could, again, I'm actually being extremely vigilant or being extremely putting a lower emphasis on the cost. It could have been, close to $100 million when it comes to drug development costs, because we were able to get the drug approved without a phase 3 clinical trial. So which again brings us back to what is the impact of model-informed drug development. So in summary, as you can see, model-informed drug development, when used effectively, can lead to saving a lot of time, a lot of money in drug development costs. And optimizing the use of model-informed drug development is strongly encouraged even by the regulatory agencies. And the way this works is you actually use the data to create certain information, and that information is used as knowledge that can be reapplied into how to optimize this whole process. With that, I think I conclude my presentation for today, and I'm going to stop sharing my slides. I know this was a lot of information that I have given you in the past 40 minutes. I would love to see if you have any questions, anything that I can help address, or any clarifications that you need.
I have a couple of questions, Aruna. One is the PDGF alpha, which is what you're measuring as a biomarker. Was there a significant difference between chemotherapy and your monoclonal antibody in the PDGF?
Yes, Nat, we did have a significant difference. As I mentioned to you, that it was actually a combination therapy. It was given in combination with chemotherapy and Olaratumab. So when we were conducting the clinical trials, part of the analysis also included that, is that we would do a head-to-head comparison between in the combination therapy and without Olaratumab, and we showed significant improvement in the combination.
Second question is from a Q3-week treatment for compliance sake, you went for Q2-week treatment. And did that have any effect on the quantifiable levels of your antibody, Olaratumab? And over a prolonged period of time, usually all these monoclonal antibodies start generating human antibodies against human antibody. Was that ever predicted, or was that anticipated in your modeling process?
Great question there, Matt. So actually, we did collect the antibody data for all the subjects on the clinical trial, and part of our exposure response analysis also included teasing out if there was any impact or a direct correlation between when do we see an antibody response and when we don't. And we were actually not able to quantify if there was a direct relationship there.
And the Olaratumab treatment is a single treatment, right? The chemotherapy was not given concomitantly.
No, it is actually a maintenance treatment. So it is multiple doses given in combination along with the chemotherapy agent. So it's not a single treatment, it's a multiple dose regimen. And that is the reason why when you saw the simulations, we actually conducted the simulations also for a multiple dose therapy.
It's not easy because the chemotherapeutic agent may have nonlinear kinetics or probable effect on the PD outcome and all those things, but still you are able to, I'm curious to know what kind of mathematical model you use, but as you said, I'll do my homework, go through the papers anyway. I don't want to hog up with questions. Any other questions from the group?
Yes, so looking at the plasma concentration curve, you were using a log scale for concentration, but nonetheless, the curves were nonlinear. So what is the nature of the underlying, is this a sum of exponential terms?
So thank you, David. Thank you for the question. What you saw in the multiple dose simulations, and I can actually bring that slide back, which would help us in understanding this better. So as you can see here, that, answering the first part of the question, that this was a multiple dose therapy, and that's the reason why you see the accumulation playing a role here. When we are moving from, sorry.
Yeah, go ahead. I'm sorry.
No, so as I was mentioning, right, that when we are moving from dose number 1 to dose number 5, you do see that accumulation has a role to play. But after dose number 5, you would see that there is a steady state. And post this, you don't see that accumulation occurring.
Right, but that should be predictable based on, let's say, the smallest rate constant. That I get, but all of these curves, no matter what the dose is, it's not reaching a log linear straight line, at least not that I can see. So what is the nature of this underlying model? Is it a sum of exponential terms?
So I think addressing your question, is it a sum of exponential terms? Based on the data that we have seen, we don't have enough evidence to say that that is the case. And in terms of, whether this was a nonlinear kinetics or not, I think that is a question that can be well addressed if we actually look at relative dose versus AUC. If we were, and in this case, again, I'm not presenting that data. However, we did do a study in which we did do multiple doses at various dose levels. Here we are looking at 15 milligrams was or 20 milligrams was the highest dose. But in the phase 1 safety trial, we did go up from 3 to 25, and that data was used to design the right dose wherein you actually don't see that kind of saturation happening.
Why are you invoking saturation? In your single dose studies, did you eventually?
This was not just, sorry. So just to answer your question, right? So this was not a single dose study. Again,
no, I understand that, but you said you said that you didn't show the data, but you had there was previously single dose data. And my question is, in that data, did the elimination phase eventually reach a terminal log linear pattern?
So first is because unlike other therapeutic areas, in case of oncology, we do have to conduct the trials in oncology patients. And we generally would not do a SAD or a single ascending dose and then a multiple ascending dose study. In this case, it was always a multiple ascending dose, and it was always conducted in patient population and not healthy volunteers. So for us, it was always more important to get to the most efficacious dose rather than getting all of the dose proportionality and the linearity and where is the saturation, etc. Focus was always about finding the optimal dose to see the maximum response.
Okay, and final question is, what is the pharmacodynamic biomarker? You mentioned some experimental animal data, but what is the human biomarker that reflects desired efficacy, if there is one?
So that was a platelet-derived growth factor alpha. That was the biomarker that was used for the biomarker determination.
I don't know if you go back one slide, that's the thing that raised the question.
Yes.
No, the one before that, single dose.
Which one are we talking about, Nat?
2 slides before.
Oh, right. Are you talking about this one?
Yes. That's what raises the question, because the vertical axis for the plasma concentration changes naturally, understandably, because of the dosing differences. But since we haven't characterized the tail end properly, it raises the question, did we reach steady state? That's the thing. Usually all these antibodies have a half-life of anywhere between 21 to 28 days, depending on the antibody. So looking at this plot raises the question, okay, you've made the simulation. Did we really reach the steady state? But your simulation shows steady state, but actually, is that what happened? That was the question that comes to our mind.
And these concentration axes in this are linear.
Yes. This is definitely on the linear scale. I agree with you. Actually, I would say, to answer your question, David, did we have nonlinear kinetics observed for this particular compound? My answer is no, not to the best of my knowledge. Does that answer your question, David?
Yeah, I guess so. I mean, I guess you don't have enough duration of sampling, given the limitations, to actually fully characterize the single dose kinetics. So there's some ambiguity.
I agree with you. I agree with you that, here we are talking about half-lives of, as Nat mentioned, like 21 hours or more. And again, this therapy is given weekly. And as you can see, based just on this plot, we're looking at long half-lives. We did not have the ability or the opportunity to really characterize the PK, because our focus was not so much on characterizing the PK, but mostly getting to what is the most efficacious concentration that would work for the subjects. I hope that answers your question, David.
Yeah, that's fine. Thank you.
Are there any questions from students?
And again, you don't have to ask me a question just based on the presentation that I have given. If you have questions in any other part, some of you I know are graduating soon. If you have questions in terms of how to prepare yourself better for the job market, I'm happy to help you there as well.
Thank you so much for your for the seminar and for telling us about your work. I have a question about, so I assume you might have had a lot of experience with all sorts of medicine to deliver to the patients, not just the cancer drugs. And I was wondering how different, or very generally, how different is the process of preparing a drug that's not for cancer patient, but something that administered chronically, for example? What are the key differences? Thank you.
Sure. So generally, drugs could be categorized into 3 categories. One is for an unmet medical need, whether it is rare disease or if it is first in class. In that case, the acceptance criteria, or what the regulatory agency is willing to live with, pardon my language there, but more so, for instance, we know about COVID vaccine development. There was an unmet medical need. It was really required and warranted because of this large scale of impact COVID was having around the world. So there was special, I would say, exceptions made by the agency. Now, that does not mean that the scientific evidence was not warranted, that was required, but just the approval process was a little faster. And in case that you have drugs which are not first in class, but there are multiple other compounds already out in the market, then the threshold that the application needs to meet is much higher. So the drug development timelines are also a little longer. And for rare diseases, I mean, there is a category that one needs to meet to fall into the rare disease case, in which case, they are less than 200,000 people around the world, if I'm not wrong. In that case, a drug that goes through approval process may not have clinical trials conducted in more than in some cases, less than 100 subjects altogether. And in my career, I have also seen that there are drugs that get approved for rare diseases wherein the clinical trial was conducted in as little as 50 subjects. So I hope that kind of gives you a little bit of glimpse of the sliding scale when it comes to drug development and approval process.
This is very interesting. Like 50 patients is not that big of a number to be very confident with the data. I don't know. That seems like it, but it's very interesting. Thank you.
Sure. And just as a side note, I also do want to mention to you that something that we have also formed is a non-profit organization called Project Dontabhaktuni. And we are focused on creating workshops and training programs for scientists and students around the world, wherein we offer workshops and training programs to individuals. I think we have trained over 2,000 PhD PharmDs from 40 countries. We do have an open YouTube channel wherein you can go learn more about these model and farm drug development techniques. We also have hands-on workshops that you can use to expand your knowledge. Currently, I think we are working on creating a Toastmasters Club, which is a public speaking club for no cost participants. And if that is something that you are interested in exploring, please follow us. We always keep presenting stuff on LinkedIn and YouTube. So, take advantage of all these free material that we make available to you.
Thank you, Aruna. Actually, if you send me some more specific links and information, I'll be happy to share them with our students and see maybe how can this type of training be better integrated with our program in drug development. So this is certainly an interesting development. Thank you.
Sure. Thank you. Happy to help. And thank you, Manus, for the opportunity to come and present. Really appreciate it.
Thank you, Aruna. Thank you for helping us with this very important course this year again. And I'm sure the students enjoyed it. And thank you, Nat, for arranging the whole thing as part of the course. And we will be in touch. If we have any follow-up questions from the students or faculty, I will let you know. Really appreciate it.
Thank you, Aruna.
Thank you, Nat.
Thank you. Thanks.
Oh.
Thank you, everyone.